Hello everyone. Now it is time to start with the next lecture, Introduction to Proteins and a Peptide Bond. Myself, Mr. Himanshu Jaiswal, I am Assistant Professor in Department of Botany, J.D. Patil Sanglodkar Mahavidyalaya, Daryapur. Friends, what are proteins? We all know that proteins are polymers of amino acids, right? They are formed by joining amino acids. Proteins are the most important and highly diverse and versatile biomolecules in the biological system. No other molecule has this much diversity as these proteins have. Due to the huge structural diversity, these proteins perform various functions. For example, they perform a function of receptors, just like rhodopsin. They will perform the function of muscle contraction, just like actin and myosin. They perform functions as hormones, insulin and glucagon. They perform functions to protect our system from the pathogens by working as antibody. They act as an oxygen carrier. They are the ones who fix carbon dioxide into in the process of photosynthesis. They are the ones who carry ferritin, that is iron, in our system, and they are one of the most important constituents of our hairs, nails, and the spider silk even. So what is protein? Protein is a polymer of amino acid which are linked to each other by the peptide bonds. There are total 300 different amino acids in the biological system that can be broadly classified in two categories. One is standard amino acids and another is non-standard amino acids. Now, what are standard amino acids? Those amino acids which are incorporated in protein. So there are 20 standard amino acids. They became a part of protein. That's why they are called as standard amino acids. On the other hand, there are non-standard amino acids which are not incorporated in proteins. There are a total 280 different types of non-standard amino acids, just like PABA and citrulline. PABA stands for the para-amino benzoic acid. Now, amino acids, amino acids can be defined as the molecules which are having one amino group and an acidic group. They are called as amino acids. So, in amino acids, there are two principal groups. One is amino group, that is NH3 positive. Another is carboxylic group, that is COO negative, right? So these two amino acids, they can combine each other. There is process of condensation, and due to this there is removal of water molecule and there is formation of one peptide bond by the loss of one water molecule. By the help of this peptide bond, two amino acids can be joined, and this peptide bond plays an important role in the determination of the secondary, tertiary and higher level structure of proteins. Now, this peptide bond has various characteristics, and one of the which is it has a partial double bond character. Due to this partial double bond character, the rotation is not possible across this peptide bond. So proteins can be rotated against only two types of bonds. One is CC psi bond and NC phi bond. So there are two bonds which allow the rotation, that is CC psi bond and NC phi bond. Peptide bonds doesn't allow any type of rotation against themselves, and that's why the peptide bond becomes planar. Now there are various forces that stabilizes proteins. The forces like disulfide linkages, that is covalent bond between the cysteine, hydrophobic interactions between the hydrophobic amino acids, van der Waals interactions between the uncharged amino acids, and hydrogen bonding and electrostatic interactions which mostly occur between the charged amino acids. So these are the various forces that stabilizes protein. In the next slide we will see how a protein is stabilized. Now, as you can see here, this is the hydrogen bond between the phenylalanine and this carboxylic group. Then there is this hydrogen bond again between these two groups, there is hydrophobic interactions between the hydrophobic chains of side chains of amino acids. There is disulfide linkage between the two cysteine residues, and electrostatic interactions between the charged amino acids. One is basic amino acid and another is acidic amino acids. Thank you for patiently listening to my lecture. I will come back with the next lecture in which we will see the structural details of proteins, what are the structural levels of proteins, the primary structure, secondary structure, tertiary structure and quaternary structure. Thank you so much.